With that, we'll turn it over to Kathy Todd Brown, and she will introduce Pat Megonigal for today.
Well, it's my pleasure to introduce Pat Megonigal, who I met a few years ago at a Coastal Carbon Research Coordination Network that he was hosting out of the Smithsonian Institute. He's the associate director of one of the associate directors of research at the Smithsonian Institute, and has PI'd way too many projects for me to go through now. I dug back into your publication history, Pat, and I noticed you have a 1988 publication on the organic matter dynamics in 4 seasonally flooded forests. And so I thought it was sort of a nice continuity to be talking about carbon dynamics in coastal ecosystems throughout that trajectory. Yeah, and with that, I think I am going to turn it over to Pat to talk about today's plant microbial interactions and the impact of homeostasis on coastal wetland greenhouse gas emissions. Take it away, Pat.
All right, well, thanks, Kathy. Let me see if I can get us shared up here. And see if that works out well. Okay. Are we seeing the view that we should?
No, we're seeing in presenter mode right now.
Really? Okay. Somehow I knew that was going to happen. Let me try that again real quick.
Excellent.
Okay. Well, yeah, I just want to start by thanking Kathy and HT Odom Wetland Center for this invitation to speak. Kathy mentioned that I have a paper that dates back to 1988 on organic matter cycling in the Great Dismal Swamp, and one of those sites was a cypress stand. In fact, it was a Fortran model that I developed. And I have still on my desk HT Odom's big book on cypress wetlands, and so between that and the fact that I was also inspired by his brother and his nephew, they were all influential for me. This is a special treat. So thank you very much for that. All right. Well, as you can see, I'll be talking today about greenhouse gases. And my particular view on this will be to illustrate some plant microbe interactions that occur really in all ecosystems that tend to impart homeostasis on greenhouse gas balances or emissions. And so I'd like to start that with a very big picture view of this big picture view. It's a graph I'm sure everybody here has seen many times, but it's showing concentrations of the big 3 greenhouse gases, carbon dioxide, methane, and nitrous oxide over the Industrial Revolution. And clearly they're going up. They're going up because the processes that emit these gases have increased more than the processes that remove them. So it's a global imbalance driving this. And this is a very simple budget for the present day imbalance of one of those gases, carbon dioxide. And what we see here is that new sources of carbon dioxide to the atmosphere in red, 90% of which are from fossil fuels, are adding about 10 petagrams annually to the atmosphere. Petagram is a very big number. We don't have to really understand it or try to imagine it here, but it's big. In black is the rate at which the atmospheric pool of carbon dioxide is increasing, which you can see is just half of the rate at which we're emitting new gases to the atmosphere. And the reason it's half is because the Earth's ecosystems are taking up about half of that CO2 each year. A quarter of that's going into oceans. That's ocean acidification. The least well-known of these numbers, by the way, is the amount going into terrestrial ecosystems. That's actually calculated as the difference of all the other numbers. And so we don't know it well, but we do understand there to be a net sink. The important point that I want to make with this graph is the size of that sink. It's big. It's significant. And this has gotten many people, including myself, thinking about the extent to which you could manage that number. You might actually be able to increase that number through management. And that is a topic that we refer to as natural climate solutions to climate change. This is a topic that has a lot of discussion right now. I chose this image from the Nature Conservancy because they've actually been pretty, they've done a nice job of trying to really quantify this, I think, in a rigorous way. And in this slide, I'm showing you some results from a paper a few years ago where this was done for the US. And all things considered, when we consider forests, aglands, and grasslands and wetlands, there is potential, reasonable potential, that we could offset maybe a fifth of US CO2 equivalent emissions through better management of these systems, which, by the way, would be good for the systems as well, not just for the atmosphere. And of course, forests are the big one here, but the point I want to make is that wetlands, even though they occupy a small area, actually rate on this graph. You can see the bar, and coastal wetlands are a piece of that. In the context of coastal wetlands, we think of these systems, we call these systems blue carbon ecosystems, tidal marshes, mangroves, seagrass meadows that occupy a very small part of ocean area, less than 1%, but sequester about half of all ocean carbon. And so the point of this introduction is to, first of all, argue that what I'm going to talk about, I think, has application across all terrestrial ecosystems and probably aquatic ones. But I'll be using, in this case, a tidal marsh and blue carbon systems as kind of the place that I'll illustrate my view of it. So the thesis that I'm going to propose for this talk is that plant-microbe interactions limit our ability to manage terrestrial ecosystems for climate change. And I underlined limit there because it doesn't prevent it. It just places limits on it. And if we understand the processes that underlie these limits, then we have a better idea of how to manipulate them. And the limits I'm thinking about in particular are basically opposing but linked biological feedbacks. So basically, we have these plant processes that tend to remove carbon dioxide from the atmosphere, which lead to less carbon dioxide and therefore negative forcing of climate. But then we have on the other side microbial processes that add these gases back. And my point is that in this case, global change is the theme of the teeter-totter there, but you could think of that as management actions that we might take. And because these things are linked, they place some limit on what we can achieve. And essentially, I think of this limit as the tendency of ecosystems to be homeostatic with respect to greenhouse gases. And here's just an example from our own work at the marsh behind me. We have a lot of experiments I'll mention in a minute. And what you see on the x-axis is that depending on our manipulation, carbon dioxide, nitrogen, or the combination of those, we see that carbon uptake or sequestration increases, which drives us to sort of a cooling, in a cooling part of this graph. But at the same time, there's a trend, it's not perfect, but there's a trend for greenhouse gas emissions on the y-axis to go up as well. And so when you combine these things, what you end up with is points falling near a one-to-one line, which is what I'm calling homeostasis. Okay, well, with that, I'm going to say something about this image behind me. This is the Global Change Research Wetland, or GCREW. On the right, I'm kind of directing you to where we are on the Chesapeake Bay in the mid-Atlantic. This site has deep organic soils, which is important because it means there's relatively mineral deposition here. It tends to be dominated by relatively few species, but these include grasses and sedges. This is important to us because the grasses are C4 photosynthesis, the sedges are C3. And I have here listed a few characteristics of the marsh. It's brackish, it's microtidal, you know, it's the area is there for what that's worth. Well, the first experiments at this site were established a long time ago in 1987. One of the very first attempts ever to manipulate carbon dioxide in a field environment started there and is still ongoing. And that is a manipulation of carbon dioxide. That was started by a guy named Bert Drake. And since then, since I joined in 2000, we've added several other additional experiments, all of which have, well, most of which have elevated CO2 as a common theme. But as we've learned something about the elevated CO2 response, we've started crossing it with other factors that we know to be important, like nitrogen, temperature, in novel plant traits, which is how we think of Phragmites invasion, sea level, of course. And what you see in the top right is an experiment we're going to start this coming summer, which I'm excited about, moving up into the adjacent forest. The ones in red are the ones I'm going to pull some data from for this particular talk. And in fact, you're going to see quite a bit of data from this warming experiment. So I've highlighted that. All these experiments are all very much deliberate, and they're all directed at understanding this conceptual diagram of the marsh, which is basically how we understand the marsh in terms of resilience to sea level rise. So we have sea level on the right here. Then we have in these red boxes elevation as kind of the key response variable that we're interested in. Elevation can increase by two processes, either an above ground loop in which the plants are interacting with mineral sediments to grow the soil surface from the top up, or a below ground loop, which will be more my focus, in which the balance between plant carbon inputs through root productivity and decomposition determine how much organic carbon is added to the surface, added to the soil, causing it to gain elevation from the bottom up through expansion. And the plants are key here. They're ecosystem engineers, and the amount they allocate to shoots or roots has a lot of control over changes in elevation. On the list on the left is a list of the global change factors that we manipulate, all of which tend to affect root shoot ratio. So that's the conceptual diagram. And I want to just go ahead and thank the colleagues that have been key to the work that I'll present today. There's many others for work that I won't be presenting, and of course funders. And maybe especially, well, all of these have been absolutely key to the work we're doing. So I won't tell you who my favorite children are right now. All right, so with that, we'll get into some data, and I'm going to present this point of view of mine in the context of three dogmas. The first of which is that elevated CO2 increases carbon sequestration. And the reason this is dogma is because we know that elevated CO2 generally increases plant productivity. And here's some evidence of this from the marsh. This is a 19-year record of net ecosystem exchange of carbon dioxide as it responds to elevated CO2 in red and ambient CO2 in yellow. And as you can see, in most years, there's more net uptake of CO2 at elevated carbon dioxide concentrations, which are basically concentrations that the whole world will be swimming in at the end of the century. And in between, I've shaded the green part, because the green part is carbon that, could be stored in the system somewhere, presumably is being sequestered in the system someplace, which would help, mitigate greenhouse gases. So if that actually works, then what we would expect based on that, the dogma alone, is that more CO2, more plant productivity, more labile soil organic matter. Now note that a lot of the soil organic matter in this system is actually old and recalcitrant. But, in an experiment like this, we should add to the labile pool initially. And if that happens over time, then we should see an increase in the soil carbon content, soil carbon stocks. However, if you add into this the possibility that that labile carbon could increase microbial decomposition of recalcitrant organic matter, then instead of seeing an increase over time, we could actually see a decrease. That is called priming, a well-known phenomena that's defined as an increase in the supply of labile organic carbon, causing an increase in the decay of recalcitrant carbon. This is something I've been interested in for some time, and I'm going to give you an idea of how we actually go about understanding problems like this. And in this case, we started back in like 2005 with a greenhouse study, small scale, something that we can do a very good job of controlling. It's Amy Wolf who led this as a technician and then published it, and has gone on and done great work. So in a controlled environment like the greenhouse, we can do clever stuff like this. We can take our sedge, and by the way, that elevated CO2 data I showed you earlier is for the C3 community, which responds strongly to CO2. But the sedge is a C3 plant and has a particular carbon isotope ratio, -25. And what we did was to plant those in soil that had been developed under C4 vegetation at our site, which has a very different isotope ratio. Then we can put a chamber over this and collect the CO2 that comes from plants in green or microbes in brown. Of course, all the CO2 looks the same to us until you look at the isotope ratio. And if we take a sample of this and the isotopic ratio is -22, we can use a 2 n-member mixing model to calculate 25% of that was from soil organic matter. So that's what we did. We did it under ambient CO2 and elevated CO2. And this is what we found. We found that at elevated CO2, soil organic matter decomposition rates were far higher, 3 times higher. So even though the plants were growing faster, the decomposers were also decomposing faster. And that's this homeostasis thing I'm talking about. Now, the default hypothesis is that this is caused by an increase in labile carbon the way I presented it. But we also observed this, that at high CO2, where we have a very high root biomass, we also had very high redox potential, a relatively higher redox potential. And this led us to propose a second type of priming, oxygen priming. Same definition, but here the supply of oxygen increases the decay of calcitrant organic matter. So we did this in the greenhouse. We actually did it in the field in an organ type mesocosm setup. But where I'm going next is sort of the most recent and grandest evidence that I think this is actually a thing. SmartX, the salt marsh secretion response to temperature experiment. At the bottom, and by the way, can you see me pointing right now or not? Okay, good. At the bottom, you see the basic experimental design, which is a gradient design where we have 4 temperatures ranging from ambient to 5 degrees above ambient and 2 in between. It's nice to have gradients like this because you can actually draw curves, and modelers love curves. Actually, I love curves. So that was the rationale for the gradient design. And then we crossed the two extremes of temperature with elevated CO2 to get that interaction. The setup is that we have this in two communities, one dominated by C3 vegetation, the other by C4. Each community has 3 replicate transects. We started the heating in 2016 and the CO2 in 2017. And by the way, this will be my last slide of the entire talk, but the whole point of this was to improve ecosystem and Earth scale models. I should point out that the scientist who's really leading this work now is Genevieve Noyce at CERC, and so she deserves much of the credit. Okay, before I start throwing data at you from this, I just want to make sure we all appreciate this equivalence that I have at the top, that elevation gain and carbon sequestration in this system, they're the same thing. Probably heard something like this from Chris Kraft. Basically, as sea level rises and elevation is gained, so is soil volume and carbon. That's the equivalency. And I want to add another equivalency, which is that in our system and many others, elevation gain is normally, but as I'll show you, not always, a very strong, has a very strong relationship to root productivity, because obviously it's the below ground productivity that's adding carbon to the soil, driving elevation gain. Okay, with all that as background, here is some data. So what we have here is on the left, on the top, root productivity is a function of temperature. And you can see that, in fact, we do see some interesting non-linearities here. Root productivity peaks at 1.7 degrees and then falls. And by the way, I didn't say this, but this system is actually really unusual and unique because we're warming the whole ecosystem. We're warming the plants above, but then we're warming the soil to 1.5 meters. So it's a whole ecosystem active warming experiment where we are adding energy on a minute by minute basis, tracking ambient conditions. So, it's very realistic. And we see this interesting nonlinear response, which I won't try to explain now, but you can read about it in Noyce et al. 2019. And as expected, elevation follows the same pattern. So that we're very consistent here. And you can read about that in a paper that was just accepted and in press at Global Change Biology by Smith et al. So far, so good. This is how we understand the world to work. But then when we cross that with elevated CO2, we see a big increase in root productivity. Okay, that's actually not that much of a surprise because we know that's a common response, but no equivalent increase in elevation at the marsh. So the only way I can understand this is if the increase in root productivity at elevated CO2 triggered a priming effect that prevented us from seeing an increase in carbon sequestration. Now, if that's the case, we should see an increase in decomposition rates. Decomposition is a hard thing to measure when you're talking about soil organic matter, but we do have some evidence to suggest that that's exactly what happened. And this evidence comes from detailed analysis of the dissolved organic carbon pools in this system. Let's start on the left, and what you see in these open box plots is the warming treatment by itself. And on the y-axis, we have something called the nominal oxidation state of carbon, which I won't try to explain right now, but the way to interpret it is that a lower nominal oxidation state means that there's been more aerobic decomposition of organic matter. And that's what we see in the elevated CO2 plots, not in the temperature only plots, but we do see it in the elevated CO2 plots, just like the pattern I showed you in elevation gain. And we see a relationship between nominal oxidation state of carbon and fine root mass, which I interpret as the possibility that increased root plant biomass is driving increased decomposition rates. And finally, is we get this graph, which is more FTCI data, but in a different form. It's a PCR plot, and you can see that the elevated CO2 plots fall on the left, which we interpret as meaning there's less lignin in the dissolved organic carbon pools under elevated CO2. So this is all indirect biogeochemical evidence of priming in the field in an in situ experiment. And but what I haven't said anything about is redox potential, because if the mechanism I'm suggesting is real, then we have to see an increase in redox potential under elevated CO2. And we finally have the data to show that that is the case. So here's 2 years of data, not the whole record, but 2 years continuous, which is, I find, fairly amazing. We have, it's only from the warmest treatments, but it's warm plus 5 degrees under both ambient and elevated CO2. So there's a couple of things to notice here. The first of all is a gut check. The redox potential at 5 centimeters in green and black is higher than at 20 centimeters in light green and gray. That is good, because if it wasn't like that, I wouldn't trust the data. The other thing to notice that there's a lot of interannual variability here. Now, this is not driven by hydrology because this is a tidal system. That means it's driven by plant microbe interactions. But the main point for us is that we see a very dramatic increase in redox potential at elevated CO2 up to 200 millivolts. So collectively, I interpret all of this as increased plant productivity due to elevated CO2 being offset by increased microbial activity. Homeostasis. All right, moving on. Dogma number 2, flooding increases carbon sequestration. The more flooded the site is, the more carbon will be stored because it's wetter, there's less oxygen. And so at our site, this is the concept that if you have, you have this marsh soil surface, the water table is say 10 meters below it. Oxygen is moving into the surface, creating an environment with fast aerobic decomposition. Below that, you have slower decomposition because it's flooded, but you do get some oxygen getting in through roots. Now, if we do something to make it more flooded, we should eliminate that surface and the high rates of oxygen related decomposition there and increase carbon sequestration. That's how most of us would explain it in an ecology class. So we did an experiment, not expecting too much out of it, just supposed to be very simple, where we had these long pipes, pots, I call them, 75 centimeters long, filled with soil from our site, planted again with this C3 sedge, but arranged at 3 different elevations with respect to the tidal creek, high up out of the water, more or less the current in the middle, the current soil elevation of the marsh, and a deeper depth, which would represent sea level rise. And in each case, we had pots with and without plants. And of course, what we expected is the highest decomposition in the driest plots. Straightforward. The first bit of data I'll show you from this experiment, which was published in 2015, is the biomass response, because that turns out to be important at the end. And in terms of total biomass, we see that plant production is actually highest in this middle elevation, that if the soil is either too dry or too wet, that suppresses plant growth. And that's a basic, very basic concept that, you know, Kathy has built into her R model, her RMM model. So, you know, this is good. And we, you know, would expect, so let's put up the next graph. Now we're showing decomposition. And the first thing to show is that, well, in the presence of plants, decomposition, and here again, we use the stable isotope trick, is much, much higher, which we had established earlier. So the presence of plants increases the decomposition rate. So that's number one. Number two is that if we look at the brown pots that have no plants in them, then we actually don't even see this hypothesized relationship with elevation. They're basically seeing the same rates of decomposition at all elevations. And if we look at the plants, the lowest rate of decomposition is at the highest pot. So the highest pot was supposed to have the highest rate of decomposition, has the lowest in the presence of plants. And the only way we can explain this is if the plants are driving the process. And in fact, we do see these relationships between above ground and below ground biomass and soil organic matter decomposition rates. And we see them if we run the experiment 1 year, or if we run it 2 years, we see the same thing. So more carbon, more plant carbon in, more microbial carbon out, homeostasis. Okay, well, my third dogma is that soil carbon sequestration mitigates climate change. If we could, whatever we can do to increase soil carbon sequestration, we're going to mitigate climate change, particularly in wetlands. And the image on the left I chose it because what you're seeing there are methane bubbles frozen in place in Lake Baikal. Methane is the source of the homeostasis I'm thinking about here. We know, or we think we know, that there is a relationship such that increased rates of plant productivity lead to increased rates of methane emissions. Now on the left is an example at a continental scale from a paper published quite some time ago. And these are non-tidal wetlands in this case. On the right is an example from the SmartX experiment that I've been talking about, same relationship at a plot scale, and one that also varies with temperature. Notice that that is a log scale. And so at +5 degrees C, we're seeing much, much higher rates of methane emissions. Now, to understand why this has to be, why this is an absolute rule of biogeochemistry, I offer you this explanation. So take a kilogram of your favorite type of dead plant biomass. Could be marsh grasses, mangrove leaves, throw kelp in there, I don't care. Once it's dead, the first thing that's going to happen is that it's going to start to decompose through aerobic respiration, a very highly efficient form of respiration that will consume nearly all of that kilogram, or in a wetland, at least as much of it as possible until the inevitable happens, which is that, particularly if we're talking about, and we are talking about soil organic matter here, the oxygen runs out. Eventually, we'll run out of the oxygen. This is a flooded site. And when that happens, that'll leave a little bit of organic matter. But when that happens, the next most efficient form kicks in, which is denitrification, but we'll run out of nitrate. But we have iron, we'll run out of that. Sulfate, we'll run out of that. And eventually we'll be left with methane production, which will never run out of substrate, because when you're talking about methane, both the electron source and the electron acceptor is organic carbon. As long as we have organic carbon there, we can support methanogenesis. But the process becomes progressively less and less efficient, leaving more and more carbon. Now, let's say we've done some fantastic thing. We've elevated CO2 is increasing plant productivity in our wetland. And now in the exact same environment, we're getting 1.5 kilograms of freshly dead biomass. Well, the system's going to run through all the same processes, end up at methanogenesis. And we will have preserved more carbon, yes, but we will have necessarily emitted more methane. It's just, so from a basic biogeochemical perspective, you cannot escape this. However, the situation is a little more complicated than I presented it, because we also have the organisms, the microorganisms that consume methane, the methanotrophs. And if there's oxygen in the system, they can actually help reduce not the amount of methane produced, but the amount that's actually emitted. So in this case, our conceptual diagram looks like this. If elevated CO2 increases plant production and labile soil organic exudates, then we'll get more methane, and we should see more methane being emitted, and we often do. But if it simultaneously increases root oxygen loss, that could enhance methane oxidation, and we could see the opposite response, less methane being emitted. Now, in most of my career, I've mostly seen the former, more methane. But recently in SmartX, we got this surprise. This top graph is just the same data you've seen before, elevated CO2 causing a really strong increase in root productivity. You've already actually seen some of this data where methane emissions in the +5 degrees warming treatment saw very, very high methane emissions. But at elevated CO2, those emissions actually decreased by almost half. So we actually saw an example of that theoretical graph that I just put up in the field. Now, when we looked at this data a little more closely, we realized that there was a relationship between the amount of methane being emitted at this site and the amount of C3 sedge biomass. This is log emissions on the Y, on the X. If the particular plot was mostly grass, it had very high emissions, and all the way on the right, if it was mostly sedge, it had very low emissions. So we went back and started looking at some of our Mesocosm data, and indeed, there's this evidence that that the two dominant plants at our site, the grass Spartina patens and the sedge Chenoplectus, just have fundamentally different effects on methane production. The grass shows, again, a log relationship here, but the grass shows what I normally expect to see, more biomass production, more methane being emitted, whereas the sedge shows the opposite. And so these are now in isolation from each other. The sedge shows the opposite pattern. And so in this paper by Genevieve Noyce in 2021, we dubbed these net reducers and net oxidizers. Basically, we're saying perhaps we can classify plants as one or the other. That would be very handy, because it is true that certain types of plants tend to either introduce more oxygen than they do, more carbon than they do oxygen on the left, or more oxygen than they do carbon on the right. And so they each have their own particular balance between these opposing effects on redox potential. And we may be able to use that to classify them in some useful way as either net reducers or oxidizers. And so we're proposing here at the bottom that we add we actually create a redox relevant type of plant trait. If plants are ecosystem engineers, then their traits are their tools. And if we can identify these functional traits that are important to models, we can advance models. And that is, in fact, where I hope all of this is going. I've been talking about one site, but in the big picture, I want to take this opportunity to introduce a new project called Compass. You can see what that acronym stands for below. The vision of COMPASS is a robust mechanistic understanding of coastal terrestrial aquatic interfaces to support models. And the figure on the right shows the point of this, that when you look at current Earth system models, they basically connect land to ocean with a giant pipe that they call a river. We know that's too simplistic and we're missing important globally relevant carbon processes when we do this. Below is what we would like it to look like, where the river includes things like estuaries, where the estuaries interact with the adjacent land and wetland and influence coastal processes that way. So that's the big vision of this new project, which is being led by Pacific Northwest National Lab. And I would say it's actually been inspired in a lot of ways from the work at GCREW that I presented. And so this is how we're trying to scale up. So just to summarize, I believe that these plant microbe feedbacks tend to maintain homeostasis on the greenhouse gas balance of terrestrial ecosystems and also probably aquatic ones. They're particularly important in wetlands because the fact that wetlands tend to be sources of methane, and methane has such a high redox, such a high global warming potential that it adds a really important element to that general concept. Now, despite these feedbacks, we know that unmanaged systems are mitigating climate change through CO2 sequestration. That's good. And if we understand those processes in detail, hopefully it'll reveal opportunities to manage this phenomena in ways that will benefit natural climate solutions. So with that, I'll say thank you and turn it back over.
All right, thank you very much, Dr. Megonigal. Please, everybody, a round of virtual or direct applause. All right. Well, that was incredible. I'm just fascinated by the duration of the experiments you've been engaging in. And I think turning some of these dogmas on their head are really wonderful, and really is the nature of science to kind of, challenge, but certainly reinforce with some pretty excellent studies. So with that, we'll open it up to some questions. And I see Christine's already got her hand up. So go ahead, Christine.
Hi, Pat. That was like really fun to see just a lesson in carbon cycling in coastal marshes. So thanks a lot. Really appreciated it. One of the questions I have is from your tracking of, these experiments over years, is whether you are seeing a broader imprint of sea level rise affecting processes at the sort of the site scale rather than the experimental scale, and what insights you're gaining from, the broader sort of transformation, if there are any, at the site that you're working at.
Yeah, well, so first of all, thanks for the kind remarks. Well, we actually have a paper that we're revising at the moment that I think speaks to that question, Christine. And I'll point out two things. One is that the sedge that I focused on so much in that talk is the most flood tolerant of the species there. And we have seen it expanding across the marsh. So we know that in general, the marsh is becoming more frequently flooded. Now, the paper that I was talking about concerns the effect of elevated CO2 on that, because about a decade ago now, maybe, we published a study that showed that elevated CO2 by increasing, which in this case had kind of the opposite effect actually of what we're seeing in the warming experiment. Elevated CO2 really was causing the marsh to increase in elevation. And we thought, okay, well, that would help it keep up with sea level rise. And that seems to be the case for that experiment. But in this publication that I'm talking about, it looks like that positive elevated CO2 effect turned off in about 2015. And actually, maybe it was 2005. Yeah, it's 2005, actually. And we were able to show that that's because of accelerated sea level rise, that basically the stress of flooding overcame the benefits, the plant physiological benefits of elevated CO2. And we now recognize sea level rise as being the dominant global change factor, the one ring to rule them all, among nitrogen and warming and so forth.
Thanks.
Kathy, did you have a question? I thought I saw your hand up earlier.
I will
Actually,
see.
I was clapping.
Oh, okay, okay.
But that's okay. I have a question sort of germinating in my head. But I think I'll hold off a bit before I ask it and make it a little bit sounder. Nice to see you, Pat.
Hi, Kathy.
There's a question in the chat from Mauricio, if someone could read that off.
Sure, I'll read it off. So Mauricio asked, you mentioned that bed level equals carbon accretion. What about sediment inputs from rivers and coastal erosion?
Okay, yeah, well, thanks for asking that question. Yeah, I mentioned that this site, because it's so high in the tidal frame, has organic soils. And therefore, our experimental data doesn't really reflect the influence of plants on sediments, but we know that those influences do exist, and we've explored those in models. When you have a lot of sediment in the system, these same plant effects that I've been talking about, for example, with elevated CO2, actually help drive elevation gain through increased sedimentation. But with respect to your specific question, even in a system that has a lot of sediment, in some ways, maybe particularly in a system that has a lot of sediment, the rate at which the surface is gaining elevation does in fact drive the rate of carbon sequestration, at least at large scales. We showed this in a paper by Rogers et al. A few years ago for a global data set. So in the big scheme of things, you can explain coastal carbon stocks in different parts of the world by their historical rate of sea level rise, which varies quite a bit from continent to continent. On more local scales, it gets more nuanced and complex. And in those cases, yes, things like watershed scale erosion rates and watershed scale sediment dynamics become somewhat more important than the actual rate of soil elevation gain. So that's my answer.
So as a follow-up comment or question from Mauricio regarding your upcoming flooding experiments, can you comment on the status and prospects of that?
Yeah, I'm happy to, because there's some good news on that. I believe the experiment that Mauricio is talking about is this forest experiment that's adjacent to the marsh. Yeah, it's a kind of a crazy thing to do, but we have these large 40 by 50 meter forested plots set up. It's an unreplicated experiment, but each plot is very, very large. And the experiment is to add on an event basis 80,000 gallons of seawater over 10 hours or 80,000 gallons of fresh water on an adjacent plot over 10 hours. And examine the response of the forest to these treatments, both in the short term and long term. And we'll do that once next summer. We'll do it at increasing frequencies thereafter over a decade. And the point of it is to try to understand these very early forest biogeochemical and plant physiological responses that are coming already with sea level rise and storm surge. So we tested that last summer. It works, and we're going to do our first treatment this summer.
Cool. Hard to know. Is that a micro, meso, or megacosm sort of experiment? It's just massive.
That would have to be in the megacosm.
Yeah. All right, now Kathy Ewell.
Okay, thanks. The question I had when you were talking about fine root production, did you say that the increase in fine roots causes more oxygen movement into the soil?
Well, that's a good question, Kathy. I said a couple of things actually that are kind of opposed to each other, but they're both true. So yeah, the root productivity will simultaneously add carbon and add oxygen. And what I've been finding fascinating with all the work that we've done over the years out here is that we actually see the balance between those two things change, and I'm not always clear why. And so we actually have two experiments that are right next to each other within meters. One, I mean, I can explain, there are some differences in their location that could explain this, but in one case, we see that elevated CO2 causes elevation gain to increase because there's presumably more carbon going in than oxygen, and the other one's the opposite. So there's some obviously important but kind of subtle things going on with this balance between carbon and oxygen being supplied by the plant simultaneously that we need to work out.
Sounds like more dogmas are being attacked.
Yeah, including my own dogmas.
I've got a quick question. How are you dealing with any sort of faunal incursions into your mesocosms and whatnot? I mean, things like fiddler crabs, and are they excluded, and allowed in? How do you deal with the inevitable variability that I'm sure they're throwing into your results?
That's a question that must come from wisdom and
experience.
There's some stories behind there. Well, we have our own story. We don't have fiddler crabs here because it's just too brackish. We do have a lot of snails, but they're kind of scattered. What we do have, though, are muskrats. And the long-term experiment that's now run 33 years, about 20 years in, one spring we walked out and there was a muskrat den inside one of our elevated CO2 chambers. And that wasn't good. So we've been trapping muskrats actually, and I mean it's a major activity, and it's probably the scariest thing for me about running this site, because everything else we have some control over. We have built it to survive hurricanes. We can do a lot of mitigation as far as that kind of thing goes. We can take out Phragmites if we don't want it someplace. But the muskrats can show up and just eat you out, like lower the surface elevation by 10 centimeters. They've done this overnight, and you're done. So anyway. That's
cool. Practical realities. Okay, we've got another question from the chat. Do you expect the same methane patterns in high salinity salt marsh where sulfate reducers may outcompete methanogens? Is this still valid dogma?
Well, I would say it is still valid dogma, but we are learning more about it, and there's a lot of nuance there, and we know that the outcome between competition of sulfate reducers and methanogens in general favors sulfate reducers, but the nuance that is coming in is that the sulfate also favors anaerobic methane oxidizing bacteria, a different group than the one I was talking about. These plant traits I was talking about can influence this, but so I'd say it's still valid, but like all these other dogmas, it's up to be knocked down.
All right. I think that's all the questions, unless anybody has one outstanding. Otherwise, another round of applause for Dr. Megonigal. Thank you.
Thanks, everybody. Appreciate it.
All right. So remember, anybody that's registered, no in-person activity next